So hello everyone. I'm Suhrid. I'm a postdoc with Cassandra here at Harvard. And no, it's not. Okay. So the general sort of questions which I'm interested in, which I think many of you are also interested in, is how do morphologies diverge? And not in far evolutionary sort of relationships, but in sort of close evolutionary relationships where things like adaptive radiation, as one of the well-known cases is Darwin finches, this kind of stuff happened, and how do these morphologies sort of diverge, where most of the context of the organism remains the same, but then there are these morphologies which look, or like, for example, here breed shapes which look very different, right? So I was trained as a cell biologist, and the way I look at it is organisms evolve because cells do, right? And the way I look at it is, if you look at the developmental trajectory of an organism, which have two, or between organisms which have two similar but sort of different in its own way, sort of phenotype, you can start to see differences emerge during development, which can be anything from a spectrum of, like, for example, in many talks we have seen things like cell proliferation, cellular arrangements, and things like that. From there to like material properties and mechanics and things like that. And these things can then sort of emerge into these different morphologies which we see in these organisms. So for that, I mean, I answer these sort of questions in a very simple model, which is a fly ovary. So for those who don't know how the fly ovary is organized, the ovary is made up of these single units called ovarioles. And these single units are like an assembly line, which basically has a germ cell at one side, and then stem cell at one side, and then has mature eggs on the other side. And the ovary is made up of finite number of these number of ovarioles. These ovarioles are set up during the developmental phase of a fly, which is the larva. And the way they are set up from this spherical ball of cell, which is called the larval ovary. The way they're set up are like this, right? So what we're looking at is the cross-section of the larval ovary as it develops. And the sort of fundamental cells which are going to give rise to this structure is these group of cells in red, which are called terminal filament cells. So they get defined as the larval ovary grows in size, and they get defined from one edge of the ovary to the other. And as they get defined, they also start stacking up on each other and forming these very distinct stacks with an uniform number of cells. Yeah, so you can imagine this thing happens in a 3D scale. So they are forming basically stacks in the middle of a spherical ball of cell. This thing also sort of scales with evolution, because in Drosophila melanogaster, you have almost all the time 9 cells inside these stacks. Whereas if you look at other species, or you sort of move away from Drosophila melanogaster, start to see more cells or less cells, depending on which species. So here, there are shown 2 species, which is Virilis and Zephyrus, which have distinct sort of number of cells. So one, Zephyrus has like 11 cells always in these stacks, and Virilis has like 12 cells. So the questions we sort of started asking is how these stacks form, because this is a terribly understudied sort of piece of tissue. So, and primarily that's because it's deeply seated within the organism, so you can't really see it from outside, and you can take it out, you can stain it and fix it, but that also gives you a very limited understanding of how these stacks are forming, right? So the second question we asked, which I'm going to address in the talk, is how is this sort of fixed cell number maintained? Why don't we see stacks of different sizes? Why do we see stacks only of one size? And how are these sizes different between this kind of different species? And sort of this work in progress, but this sort of trying to attempt to understand these 3 basic questions. So like Stefano said, if you put in enough effort, you can grow everything in the dish. So what are the first things which when I came to the lab, we started to do is try to recapitulate the stacking process on a dish, right? And here you see these 4 cells, which are at the start of the video. These cells have been differentiated to form stacks, but they haven't formed stacks. So between getting differentiated from stacks and forming the actual stacks, there's a bit of a time lag. And we can recapitulate at least part of that process in vitro, or ex vivo. This is how you sort of see, and this is a maximum projection. So what you're seeing is a part of that ovary. And then you can see them neatly stack up on each other while other cells are moving basically around, right? And this is how the stack looks. So they're not epithelial, they're highly like fibroblast-like cells, which they're mesodermal in origin, and they sort of stack up on each other like pancakes. Right. So one of the first things we obviously thought is there some sort of a cell sorting thing going on there? Because you see these cells then sort of jam up on each other and other cells are moving around. So we looked at EK-driven levels, right? And what we see here, what you can see here, is this part marked as is a stack. You can see there's a lot more EK-driven between the cells which are stacked than the cells around it, right? So is it then cell sorting, and then can just having sort of a differential addition explain the formation of the stacks? Even though it's Drosophila, it's kind of hard to knock down EK-driven in this system, because as you might know, there are these Galford lines which have leaky expressions everywhere. And EK-driven is such an important gene that if you even have a leaky expression somewhere else, for example, in the neurons in the brain, you're not going to have a flag. So that has been a bit challenging. So with my colleague Chandra, who's a biophysicist, we started sort of playing around with a toy model, which is based on the cellular pots model system, starting with a very basic sort of assumption. So the assumption is that obviously these cells have differential adhesion, so they are more likely to sort of adhere with each other than with the surroundings, which we model here as matrix, right? And what you see is this. So you see them sort of attaching to each other from like forming clumps, but as it should, and as differential addition should give rise to clumps, essentially, which are not really stacks. So what we did is we introduced a sort of shape constraint, right? We said that, okay, if this shape constraint exists, do we form a stack? And we sort of did form a stack in this case. But what it really helps us to do is to understand that how this, or sort of find an experimental point of reference from which we can start asking questions. So this is how the stacks look like when they are kind of formed. So what you see in magenta here is active. So these cells, they're basically jammed with each other, and there's a huge sort of concentration of actin which is around these cells. And then you can see that they're sort of oscillating more or less, but they're kind of holding each other together, right? And so what we sort of, and that's where we thought that the sort of that shape constraint comes from. So we tried to remove that shape constraint in this model from these already stacked cells and see if they sort of fall apart or not. And you can basically see that this process, if you remove the shape constraint, it sort of goes back forming a clump, right? And then we tested this on these stacks with the help of cytokinasin. And what you see here is cytokinasin is a depolymerized actin, and as you depolymerize actin, these sort of cells break apart from a stack and then form a ball. Right. So we also wanted to figure out which way the sort of force or tension works in these stacks, because there are different ways to stack something, right? So if you stack it and if you're holding it down at the bottom, there's going to be a force dissipation which works. Yeah.
Can I ask a question about the cytokinasin D experiment? do you ever see in those experiments cells, like, do they always form a clump, or do some of the cells, like, do you ever see a cell migrate away from the clump?
Yeah, so it's about time scales basically. So if you look at like in a couple of hours or so, you always see the form a clump. But we haven't really checked at what happens at a longer time scale. It might slip away in a longer time scale, but their cells are not very happy with cytokines in general. So that's one of the experiments which has been on our list, to wash off cytokines and see what happens. Yeah.
Yeah, so striking is this actin H that you have around it.
Yes.
Is this one cell? Or is this in all cells, everybody contributes something to the cage?
Yes, everybody contributes something to the cage. That's the short answer. But they're not sharing surfaces. Each cell is basically just shares the surface with another cell. They're not all sharing surfaces. They're truly stacked in that sense. There seems to be higher concentration of actin in the middle, and then we will see why that is, or what that might cause. But we don't know why sort of the foci of that actin is more towards the middle. We don't know why that happens. Right, so we wanted to figure out the direction of tension in these sort of stacks and clumps. And what we did was went and did sort of ablated these cells. But you have to think about laser ablation in terms of a highly 3D tissue. So it works very well when you have flat epithelial sheets. You can cut, you can infer forces from retraction, from relaxations. But how do you do that in a flat cell, right? Along 3D. So what we did instead was just went and kind of did a point and shoot sort of experiment where you just try to kill one cell and see how the others react. So when we did that, basically pop out these cells. They don't pop out, they basically collapse. But when you sort of see the immediate fast time scale movements along, which is also shown the diagram here, as you can see that it's sort of, like Sebastian was asking, that the foci is more towards the middle, and these cells are basically kind of clumped to each other. Right. So we also found that there are some sort of molecular correlates of increased mechanical stress in these cells. Yeah.
Sorry if you said this, but earlier when you showed the video of the cells moving around and then eventually forming the stack, at that point when they start doing that search, is the number of cells that can form a stack already defined, or is that not yet?
Not yet defined.
Okay, it is not.
Yeah. So there seems to be a process where there's sort of a seeding at first, like there's a group of 4 or 5 cells which form the first stack, and there's more and more sort of cells joining up on the top. And that could be a driver of why that foci is sort of in the middle, because if you're starting off on the bottom and then you're joining more cells on the top, sort of the place where everything is held together shifts from bottom to the top. Yeah.
Can you go back for a second? So in this, where you do the ablation, it's the sort of the issue that you've killed the cell, but the cell is still there. Yeah. And so how do you think about that? Because it looks like the cytoplasm is still there. The nucleus falls apart.
Yeah. So what we are trying to do is we're trying it different ways, right? So we're trying to go at the nuclei. Okay? And for reasons which become obvious later, we also learned that the nuclei is kind of one of the things which is holding or giving shape to the cells in some sense, right? So the idea was to basically knock out the nuclei, then we get a little bit of relaxation, but like not a lot. That's why you don't see a large scale movement, and you see a very fast movement at a sort of a very small spatial scale. So you don't see a huge movement. But you can still see that the dark part is kind of still there, right? Because you're not removing the volume of the cell. Yeah. Right. Okay. So stresses, right? So we looked at these molecular sort of correlates of what the stresses might be inside, or is there an increased stress inside? And we found that these stacks show a sign of increased stress, which is mechanical stress, which is apparent from the increased amounts of intensities of acylated tubulin inside these cells, and also increased number of these strands, and also lamin AC, which is kind of a mechanosensitive lamin or a nuclear enveloped protein, which is sort of expressed at high levels, or like sort of localizes to the nucleus at high levels when the cells are under some sort of mechanical stress. Right. So given that there was a sign of mechanical stress upon the formation of a stack and not in the cells before, we wanted to look at how these cells look like before and after the form of the stack. So we went in and looked at the sort of 3D segmented volume of these cells. As you can see here, the cells, when they're not stacked, they're kind of elongated, big and elongated, whereas when they're stacked, they form these sort of disc-like shapes, which are then very roundish and sort of completely, sort of more flatter than, I wouldn't say flatter, they're more disc-shaped. Right. So, and also we looked at what happens to the volume, two basic parameters, which is the volume and the surface area. And what you can see here is that the cells are kind of more elongated, so they have a higher surface area to volume ratio and also a higher volume. But once they form these stacks, they undergo a change in shape, which is apparent from the surface area to volume ratio, but they also undergo a change in volume. So there's a volume compression happening when they are stacked. Right. So we went in and tried to see that if there's a ways which we can change the stiffness of these cells and find something useful which will let us know how these stacks are formed. So this is how it's control conditions, and this is how the cells look like. And this is just a representation of the top cells. And in under control conditions, you have about 9 cells per stack. And this is shown here. The first sort of genetic trick we went to is knocking down this protein called misshapen. Misshapen is one of these proteins which phosphorylates mucin, and it's one of the proteins which also is known to control particle rigidity and stiffness in Drosophila. So we knocked that down, and we only knocked it down just during the formation of these stacks. So there was no change in initial conditions. Just when these cells are differentiated, they're supposed to form these stacks, and then you knock these proteins down, and we see what happens to the stack. So we looked at the individual cell shapes, and the cell shapes are completely wacky. We're still trying to understand how to interpret it, but what we see is essentially these long filament-like or sickle-shaped cells. But what we see at the level of the stacks is we can now get more cells into a stack, which is also sort of shown here. And this happens over and over. But what you also see is that the variability now increases, right? Because you have less control over the stiffness, you have bigger variability on how many cells you can have in the stack. Yes.
Are there fewer total stacks?
We're actually quantifying that right now. So from the larval stages, we don't see a change in number of, we don't see a big change in number of stacks, but we are doing it at scale right now to basically have a distribution for each ovary, essentially. And and there's also some, because of these sort of wacky geometry of cells, sometimes you have 2 stacks fused into one. Because the cells are now so sort of less stiff, it can span stacks, basically. A cell can start off at one stack and then fuse to another stack. Yeah. Okay, so then we also did an overexpression, which is known to increase stiffness, and what we get are these big round cells, which are also end up having less number of cells in the stack. And this is something we see also in the distribution. But all these cases, you might notice that the variability also kind of increases. So yeah, for the fourth condition, what we did was, as telling Deepa about it, that we increased the nuclear stiffness, because what we see is that there's a big amount of deformation of the nuclei once the cell is stacking. And the idea behind this, and this has been done by others as well in the blastoderm, is that if you make the nuclei more stiff by knocking down one of these nuclear laminar proteins, you resist the change in shape in the cell. That's what we see in our case too. That can be this bulge you see here is basically just the nuclei which hasn't deformed, and this is how the cells look. And you see this is the nuclei, and this is how the nuclei completely look. So they haven't basically deformed. And what you end up seeing is that you also end up putting less number of cells in stats. Right. So we wanted to see what are the cell level parameters which are changing. What we see is that in a normal control situation, you have a certain volume and a surface area. But when you knock down when you decrease stiffness, you end up with a higher surface area to volume, so that you spread out the cell more, but you don't change much on the
I guess how about thinking of like the overall packing of the stack instead of individual surface area volume?
So you mean the volume of the overall pack?
Yeah, because you see them either lined up or these interdigital.
Yeah, so we haven't done. So I think volume of the overall pack is pretty vague. Okay, and we haven't. We probably might be able to see some patterns if we do in hundreds, right? So if we do hundreds of these stacks, which means hundreds of segmentations of these stacks, we might be able to see some pattern. At this point, we have tens, we have like 20s and 30s, and we don't see any specific pattern with the total amount of.
Like not in these conditions because it looks very different.
Yeah. So you're saying about, are you talking about?
Just the overall shape, surface area to volume of the stack, thinking of it as a packing configuration. How well are these individual cells packed when their stiffness is changed?
Yeah.
In these different conditions, and then thinking about variability instead of individual, because the thing that you care about ultimately is total stack, right? Not individual.
Yeah, I mean, yes. The thing which I care about is the how many cells go into the stack, so that you eventually end up with different number of stacks.
Okay.
That's what I care about. But I mean, you're right. I could look at the stack sort of geometry, in the sense you're talking about like developing some sort of way to look at packing, like how well they're packed. Yeah, we can talk about good ways to do that. Yeah, I think. Yeah. Okay. Right. So these are the associated changes which you have on the cell shape, on the cell shape and size. And when you increase stiffness, you don't change sort of the surface area to volume ratio so much, but then you increase the volume a lot, so they don't compress. And the same thing happens when you increase stiffness in an independent way by making the nuclei more stiff, is that you also sort of converge on this kind of volume, but you also don't change surface area to volume ratio so much. Okay, so now, so the next question we asked is whether this happens in nature, in the sense when we see these different species which are packing different numbers of cells, do we see an associated change in the volume and surface area to volume ratios of these cells? So this is still work in progress, but yeah. So these are how the individual cells look. It's not very intuitive from looking at them how they might be able to pack more or less cells, but we can look at how the surface area to volume ratios and volume changes. And what we find is that melanogaster is sitting somewhere over here, and in Zapier, which is packing about 11 cells, we see a we see a dip in the volume, but not a big change in the surface area to volume ratio. Whereas in Viridis, which is packing 12 cells, which is about like three or four more than what melanogaster is packing, we see an increase in sort of little bit of both, right? So we see, we see a decrease in a little bit of both. We see a decrease in volume and also an increase in the surface area. So they're spreading a little more. Right. So what do we understand about this? So for now, we understand that this stacking involves some sort of an anisotropic deformation, which is basically both a change in shape and a change in volume. We also see all of these internal sort of arrangement of the cytoskeleton change once they're stacking. We also see that if we mess around with either surface area or volume, we can change how many cells go into these stacks. And looking at if the way that's has happened over evolution, we actually find evidence for using both change in volume and both change in surface area or volume. Yeah. So that's pretty much it. Time. Okay. If time permits, maybe you have questions. I can take questions now.